So chapter 3 is lab tools, and this is the methods to study organisms, including the media, the nutrients, and different things. So the first thing is how to get a specimen in the first place. I need to get a specimen of microorganisms. How do you get it, and where do you get it from? You can get a specimen from basically anything. You can get it from body fluid. I can get it from the blood, I can get it from the urine, I can get it from the feces, I can get it from any body fluid. You can get it even not from fluids, you can get a sample from the water, you can get it from animals, plants, volcanoes, you can get a sample from anything. And you're going to study it. But the way to get that sample, there are special tools that can be used for that purpose. And you will see that these tools that can be used. So how to do a culture? We are doing a culture. What's a culture? You take a specimen from that contain organism, and then you will put it on a medium. You allow it to grow. You study it. You identify it. Okay, like you have certain disease, whatever it is. We are suspecting something. What exactly is going to happen? We are suspecting meningitis, for example, or whatever it is. What are you going to do? Okay, from taking that to put it in the medium, a special medium that will allow it to grow, and then you observe that, you look at it, you inspect, and you know what it is, you identify, and you tell exactly what is this. Okay? So we call that the 6 I's, and this is very important to know. So we will go through the details, but the 6 I's are inoculation, incubation, isolation, inspection, information gathering, and identification. All of them start with I. We will go through them one by one, but this is the way from the start to the end. From taking the sample until you give a result, until what is this? Basically, inoculation means to inoculate something, meaning to get that sample of organism and you put it into a medium. Okay, this is inoculate, so introduce it into a culture. Incubation, on the other hand, is when you incubate something, like we say that the baby is premature and we put it in the incubator, right? What's an incubator? It's something, a media that contain everything needed for that baby, right? Same thing here. You put it in a medium that contain everything meaning everything, including the temperature, including the amount of air, oxygen, everything. And we'll go through that. And then you're going to isolate if it is more than one species, and then you inspect it, get the information, gather it, do the analysis, and at the end, which is what we actually need, and that's why we're doing all of this, to identify. Tell me, what is this? So the first thing is inoculation. How to inoculate? Inoculation, again, to inoculate means to get a sample and put it in the medium. Okay? How to get it? You take it from anything, and I mentioned that before, right? You can get it from anything. Body fluid, body tissues, anything, animals, plants, volcanoes, water, anything. You can get it from anything. And you study it and you will know what kind of organism is there. So the sample, you're going to put it into a medium, a container that have the medium, and this medium is supporting the growth. And we will see exactly the details, but basically we know that this medium contain what this microorganism need to grow, or this group of microorganisms. The medium, and we will study it in details, can be solid or liquid or semi-solid. Solid, semi-solid, or liquid. Solid and liquid are the most common ones. Semi-solid is not that much. Where are you going to put it? You can put it in a tube, a plate, a flask, eggs, anything. Most likely it's plates, but we will see. How to take that sample in order to put it in the medium. You're using one of those, a loop A loop is something looks like this. Okay, take it like this and you put it there. Something like a spoon, like the Q-tip, something like this. A needle, just a needle. A syringe, the whole syringe. Like this is a syringe. This is a needle. Swab, something like the Q-tip. So any one of those can be used to transfer and to inoculate. And the next step is to do incubation. When you incubate the organism, as I mentioned, meaning you put it in an incubator. What's the incubator? An incubator is a media that is controlled in every aspect, all aspects. You control it. You control the environment. You control basically the temperature of that incubator. You put the media that's needed, or the nutrients that are needed to grow the organism. And then you leave it there. How long should you leave it in the incubator? It depends on what are you studying. There are certain species that will take hours, a couple of hours, and that's it. Others will take days. And then you are going to look at it. Look at it with your eyes and see that changes happening. So I put like in a tube. I'm using, you can use a tube, you can use a plate, you can use a flask, whatever it is. So you can use whatever you want. If you got like a tube, okay, a test tube, and you put the sample with the nutrients, you put it in an incubator, and you know that for this organism to grow, I need the temperature to be 60 or 70 or whatever it is. Okay, and I know these are the nutrients needed for this organism. How to know that this is the time, if the time is up, it's good now, you can go ahead and move on. It's the visible changes. So you put it like it's purple, right? Clear purple. And you put it there, leave it for whatever, we should know, or they know the exact time that's needed approximately. So we're going to study it after a couple of hours, after a couple of days, whatever it is, depending on the species. And then we look at the changes. It was clear purple, for example. Now it's getting a little bit cloudy and it's getting a little bit solid, and you see patches or something. So these are visible growth changes. These are changes that are visible, and you can see it and you say, okay, I'm done now. I did the incubation. Okay, so incubation, most importantly to remember about it, controlled temperature and nutrient factors. This is the most important thing, okay? The incubator is to allow the growth of the organism in incubator, which have growth factors and control temperature. Okay, this is what we need to understand here in the incubation. So you're done with the incubation, you put it there, you put a control temperature, you put the medium, it's growing, you looked at it, and it looks like it is okay now. So what should you do next? You are going to isolate the organism. The newer techniques, so let's say I got a sample from the urine or something, and you put it in that medium, you look at the changes, you can tell that it is changing, so you're done now. And then you're going to isolate the different organisms. However, the new techniques that are used in the beginning from the inoculation, you can inoculate different organisms, different spots. So you can put one organism here. Now you don't need the isolation. It's basically you are putting one type of organism. I got this Staphylococcus epidermis. I put it in that medium to grow, and that's it. So you don't really need an isolation. Okay? This is excellent if we can do it. Most of the time we cannot do it like that. But if that is done, you call that a pure culture. So what's a pure culture? A culture that contains one species only. Clear? If that's the case, you're done with the isolation because you didn't need to isolate in the first place, right? You inoculate, and this is not easy to do. It's kind of complicated to do in order to get one organism and put it in the medium and allow it to grow. And this is the only one that you see. This is called a pure culture, which is not that easy. Most of the time, you have a mix. And you need to isolate. So far, we didn't need to isolate, right? It is already pure culture. Did we get what a pure culture is? One species only. already isolated. You don't have to isolate. What if that's not the case? If that's not the case, you have a mix of organisms. What are you going to do now? You put it in the nutrients and you allow to grow until it looks like a mound of cells. Mound is looks like a circle, a big circle raised over the surface. And when you see that, you will see the cells are growing like a colony or colonies. The colony is one species. So what's the difference between a colony and a pure culture? Pure culture contains one species only. A colony is one species within the nutrients. So I can have this plate, and I have this colony, and this colony, and this colony. Each one colony represents one species, but you have multiple species. Is that clear? Did you get the idea? So if that's the case, so what's a colony? The colony is one species only. What's the difference between that and pure culture? You have several colonies. Okay? Each one colony represents one species. How are you going to isolate if you need to? There are 3 different types of plates that are usually used. And you need to remember, most importantly, the name of these plates. Okay, so how to isolate? I put it in plates. What are these plates? 3 types of plates. Number 1, streak plates. Streak plates, this is a technique that's using a plate and then you streak in that plate. So we call it the streak plate technique. In this case, you will need inoculating loop, the loop that you're going to use in order to make these streaks. The best way is to sterilize first. They do it like this. They get it, it's a metal, they get it, they put some alcohol on it, they put it on the flame just for a few seconds from far away, and then you return and use it, it is sterilized. Why are you doing that? You don't want to mix it with something else that you introduced, right? So you re-sterilize first, and then you do the streaks. Look at this picture right here, or these. This is the streak. So you get it and you start to streak like this to separate them apart so you can see the different species. The second type is called pour plate technique. And from the name, you're just pouring the organisms. You pour it in different spots and it's going to distribute. There is a YouTube video here that's describing this. This is the pour technique. Just look at it. You're pouring it into different spots like this. So it is spreading already without needing to streak. You just pour it. Just mix it like this, and then you start to study it. So this is the pour plate. You pour it into different spots. And the third one is called spread plate technique. This is where you spread it from the beginning instead of streaking. And this is showing you what is this. This is the flame. You are going just to re-sterilize before spreading in the plate. Here is the plate. And this is the sample. And you're going to take the sample and you're using this. It's called the hockey head tool. Put in alcohol, sterilize, re-sterilize, and then you're going to spread using it just like this. The next step is to inspect. If this is a pure culture, you have one organism only, this is easy. You are going to inspect it, you look at it first, and we will see the details, look at it, see how it looks, what changed. And then you can look under the microscope and tell exactly the details. This is if it's a pure culture. If it's a mixed culture, this will be something different. You need to recognize the different colonies. Okay, so here is one thing that's interesting, and you need to understand it, which is, what if I'm doing a pure culture, right? Pure culture means one species. So I get some Staphylococcus epidermis, for example. I put it into that, whatever the way spread or streak or what, pouring or whatever it is, and then I put that Staphylococcus epidermis, and then I look under the microscope, it's inspecting, and I found Staphylococcus epidermis and E. coli. What does that mean? It's contaminated. So like I mentioned that the tools that you're using, you're supposed to sterilize. So I take that Staphylococcus epidermis, I put it in the nutrients, and I'm looking at it, I see two. You put one, or it comes from the air or something. So if you are using a pure culture, you're supposed to see one species only. If you find more than that, translation, contamination. It's contaminated somehow. So what do you mean by inspection? And then you will do information gathering. You do two things: macro and micro. Macro and micro. Macro means just observe, look at it. How does it look like? It will look different when you look at it with your eyes. You see the growth characteristics. It's a circle, it's growing, it's cloudy, it's this, and the color changing, whatever it is. This is the macroscopic way. Just look at it and tell from the characteristics. And then you'll take a sample and put it under the microscope. When you put it under the microscopes, you can see the details, including the cell type, what type of cell, including the shape, how does it look like, and you can see the rest of details. You can use that, you can do that with or without needing a stain. It depends on what species are we talking about. Some species, you do macroscopic, look at it, put sample under the microscope, look at it, what cell type, and what's the shape. And you get the details, but others will need staining. And if that's the case, you use a microscope, a special type of microscopes to identify this. How to identify the microorganism? Identification means you need to give a name simply. You need to give a name to that microorganism. I need to exactly know what it is. How to know that? It's a lot of details. So you're going to put a name or identity to the microbe or the microorganism. You are going to gather the information that you got. First, from inspection. You looked at it, and I'm telling it's cloudy, it's growing like this, the color changing, whatever it is. This is one. And then you do some identification. And then whatever the information that you gathered, macro and micro, all this information, you are going to put it into a computer that have programs that's going to analyze. Did you get the idea? So you put all this information into the computer, you enter it. I put the organism in this of this sample. It took me this hours. It looks different. Here is how it looks different. 1, 2, 3. And then the cell type is this. The shape is this. The wall is this. And you put all the details, and the computer program will do the analysis, and it will tell you exactly what it is after you put enough information that's needed. How can you tell? To be very specific, because according to what you're doing, you are going to develop, like if this is a patient, you are going to develop a treatment plan, right? So it has to be very accurate. You need to know exactly what it is. In order to be accurate, there are different ways. You can do the morphology. Morphology means the shape. This is the first thing. Cell and colony shape. How the cell looks like, how the colony looks like. This is the first way, which is what we talked about. Macro and micro. In those two cases, it will give you an idea about the morphology of the colony itself, which is something that you can see. I look at the plate, and it's getting bigger, it's getting cloudy, it's white in color, for example. You call that the morphology of the colony itself. And then you look at the cell, give the details, tell me how the cell looks like. This is the first way. The second way is, which is more complicated and more promising, and this is a newer technique that's used, which is the DNA sequence. Instead of counting on your eyes, whether you're looking with your eyes or looking under the microscope, you do a DNA analysis or DNA sequence, and this is more accurate, more promising, more advanced. You can do both of them. The third way is biochemical test. You can study the organism and see the chemical and metabolic characteristics. So you're getting into more details. You're studying the chemical changes, the cycles, the chemical cycles that's happening, the reactions, metabolism. This organism is using glucose and it's producing this and that. You're studying the chemistry and the chemical and metabolic characteristics of that organism, and we call that biochemical. Bio means the organism, chemical means the chemistry of that organism. The last one is to do immunological test, and you like putting a treatment of certain way and see how it is working, extract antibodies or immunological test. We'll go through the details, but you need to remember these 4 ways to identify. Okay? Number 1, morphology, DNA sequence, biochemical techniques, immunological tests. You should remember this. So all of this, these are the steps to tell what is this. Okay? And we talked about, we said you put it in a nutrient medium, that's a special medium that we know that will allow the growth of this organism. Okay? How about the nutrient itself or the medium itself? The medium can be from the physical state, can be liquid, semi-solid, and solid. What's the most common? Liquid and solid. Semi-solid occasionally. Okay? It can be one of the following, according to the chemical composition now. So according to the physical, you have 3. Solid, semi-solid, and liquid. This is the physical differentiation. The chemical differentiation, we have different ways or different types according to the chemical composition. It can be synthetic or unsynthetic. Synthetic means chemically defined. You made it, you know exactly everything in that medium, right? Synthetic means you made it, right? It's not like you're taking nutrients from broth or a meat broth or something and you don't know exactly what it is. No, synthetic means you know exactly everything in there. All the numbers are known to you. Or it can be complex, means you don't know everything about it. So according to the physical states of the media, it can be liquid, it can be solid, it can be semi-solid. Liquid is like broth. And remember that the liquid does not solidify. It will not become solid. Okay? Like broth. You get a broth, meat broth for example, and you put it as a source of nutrients to the organism. You put the organism on it, that's it. You cannot change the physical state. Semi-solid and solid, they can change. Semi-solid will contain solidifying agent, so it can change to become solid. If it is solid, which also contains solidifying agent, but it is firm to start. So what's the difference between semi-solid and solid? Both of them contain solidifying agent, okay? An agent that makes them solid. or can make them solid. What's the difference then? The semi-solid is, to start, it's semi-solid. The solid is firm. The surface is firm. So it will make the colony easier. It's a firm surface. Solid can be liquefiable or not. We have different types. Okay? This is liquid and that's it. You cannot solidify. This is semi-solid, you can. This is solid, it's already solid, it's firm. But you can liquefy or not. We have different types. So mentioning the solidifying agents. The medium, some of these media are semi-solid and contain solidifying agents. And the solid contains solidifying agents. What's a solidifying agent? An agent that can change the physical state into solid. Agar is the most common one. The agar is the most commonly used solidifying agents, and they get it from the red algae. Remember it? Agar, algae. Agar, algae. Okay? They get it from the red algae. It's basically the most important component in that agar or red algae is the polysaccharide. Okay? It contains polysaccharide. So what do you mean by solidifying agent? What exactly is it going to do? Why are you adding that to the medium? Agar is solid in room temperature, but you can liquefy and you can solidify. You can change. The numbers here are important to remember. This is agar. I add it to the medium. What exactly is going to happen? Can I play with the physical state? Can I change it? Yes, you can. How? If you increase the temperature to 100 degrees centigrade, okay, it will become liquid. And if you put it towards 42, it will become solid. So under room temperature, it is solid. Okay? It's in room temperature, solid. Can I liquefy? Yes, put it in 100. Can I solidify again? Yes, put it in 42. So these are three: room temperature, 4200. So what's the importance of that agar? It provides a framework to hold the moisture and the nutrients. So the moisture will stay, like the status is this, it's semi-solid, it is solid, whatever the status is, it's not going to change. Okay? You're not losing the water, for example, of that medium. So it acts like a framework, like you're putting a cover on something, so the water does not evaporate. Hold the moisture and the nutrients, so the nutrients are not going anywhere. But it does not have nutritional value at all. So is the agar nutrient? It's not a nutrient at all. All what's doing is changing the status, the physical state. That's number one. Number two, it put like a framework to keep your moisture and keep your nutrients. Is that clear? Where did you get it from? Red algae. What's the nutrient value? What's the composition, the main composition? That's not a nutrient, but just in the composition. It's basically polysaccharide, but it's not useful. Okay? It's just part of the characteristic. You get it from red algae. Remember 142 and the function. The types of media that can be used. There are a lot of them, but the most commonly used or the most important are the nutrient broth. Is the broth liquid or semi-solid or solid? Broth. It's liquid. So nutrient broth is liquid, and it's usually beef extract and peptone. The second one is a nutrient agar, and this is solid. It also contains beef extract peptone plus agar, and that's why they call it nutrient agar. Okay, and where do we get the agar from? Red algae. Okay, so nutrient agar is an example of solid, nutrient broth is an example of liquid, semi-solid is not used that much, but it still exists. So this is very important to know. The chemistry of the medium. What are the different types from the chemical contents? The media can be synthetic. Synthetic means it has organic and inorganic, pure organic and inorganic compounds. This is your clue here. Exact chemical formula. Exact chemical formula. I know that I have 62% of this and 18% of this and 50 milli equivalent of this. You know exact composition. And it's because it's synthetic. Didn't you? You are the one who made it, right? So you control everything and you know the exact chemical structure. Okay? The second one, which is not synthetic, so you don't have much say on it, we call it the complex. And this is the one that contains at least one ingredient that you don't know what it is. It's not chemically definable. Is that clear? If you know every single detail, you made it, and you know all the details, exact numbers. And also, add to that, and we will see it, reproducible. So I can make 100 of those, I can make 1,000 of those. All of them are copies of each other. Is that clear for the synthetic one? Synthetic one, you know everything about it, and everything is exact numbers and reproducible. You can make a lot of these. Complex, it's not synthetic, so you don't know exactly what everything in there. At least one ingredient, it can be more than that you don't know. Like you study it and you know 1, 2, 3, 4. I don't know what is this. We call it complex. The third one is general purpose medium, and this is one that's that can grow a lot of organisms. Obviously, that's not synthetic because you can put any organisms on it and it will grow. And from there, you can determine the next step. Either that's enough or you put it in another medium. Number 4 is enriched media. Enriched media is the one that's enriched. Enriched means it's rich. Enriched, it's rich of nutrients. Like you get it from the blood, serum, hemoglobin, something like that. And not only this, but you add growth factors to it. It's called the enriched medium. This is if you need to do a fast growth of the organism, okay? And you use that like, here is a patient that's in critical condition and I need like half an hour and you tell me exactly what it is. You cannot wait for the other media, right? The other media you can wait hours and days, right? You don't have that leverage. If you need to grow it really fast, what should you do? Enriched media. Okay? This contains the organic substances, basically. They get it from different sources, but at the end, high nutritional value, and you add growth factors to it. Is that clear? When do we need the enriched media? If you need to grow something very fast. You have an hour and tell me exactly what kind of organism do we have here, or the patient will die, for example. Other types of media are selective and differential media. Selective media is a media that's going to, this is the simple way. Selective media, a media that selects what to grow and what not to grow. Selective. How? Nutrient for this one, inhibiting factors for the other ones. Okay? Example. I need a media to grow E. coli. Okay, are we following so far? I need a media to grow E. coli. And I get a sample that contains different organisms. When you put it in that selective media, what's going to happen? This selective media for E. coli, it's labeled E. coli. So selective media for E. coli, what's going to do? even though you're adding a mix of culture, right? What's going to happen? This media contain growth factors for E. coli and inhibiting factors for anything else. So when you put the sample there, 3-4 types of organisms are going to die, are going to be inhibited. And you're promoting the growth of the E. coli, for example. This is called selective. So to remember it, it's select what to grow and what not to grow, simply. The next type of media is called differential media. And differential media, it will to remember it, it's the one that will show you the difference between organisms. Differential difference. Differential difference. So it show you the difference. This is a media that is produced in a way to allow you to differentiate. Like look at this picture right there. You will see red, purple, green. Each one of those is growing in a visible way. So you can differentiate. You can see all three and you can tell this is number 1, this is number 2, this is number 3. So this is differential media, and your clue is difference. Differentiate. Difference. Will tell you the difference in a visible way. Okay, can you answer this question? Irrespective of the name, just read the rest. So what is it? Differential, yes. See how it's giving you different shapes? You can tell the difference visibly. It's visible change. So yes, this is a differential one. Okay, so this is for chapter 3.